chapter 8. All right, so we're just going to do a preview of chapter 8 and not a huge in-depth discussion, but I'll tell you what things that I want to emphasize. So Mendelian genetics is not the whole of genetics. Gregor Mendel is this guy holding the pea plant in the back row. And he had a lot of time to study science at his monastery, and he studied pea plants because pea plants don't run away and they grow quickly and they have a lot of offspring, all those pea seeds. And in the several years that he grew and studied patterns of inheritance in pea plants, he identified two laws of inheritance, the law of segregation and the law of independent assortment, that are still relevant and valid today. When he was doing his research, he didn't know anything about chromosomes, or DNA, and how those were inherited. Anything that Darwin did, anything that we knew about mitosis and meiosis, that all came later. And yet he was still able to figure out patterns of inheritance. But there are a lot of things to notice about pea plants, and he settled on 7 heritable features or characters that he could determine were inherited separately. And we now know that those 7 characters were on different pea chromosomes and not traveling together on the same chromosome. He ignored the things that were inherited together because he couldn't figure it out. And when I say each character had 2 versions, think about two ways to do things, right? If there was only blue or green eyes, then there would only be 2 gene options for eye color. Well, he was looking at pea plants that had only 2 gene options for all of those characters. And the next slide that I want to look at here is this one that shows the 7 characters. So flower color, the flowers of the pea plants were either purple or white, nothing in between, no other color. The flowers were either at grew along the sides of the stalk or grew at the end, which is what we call terminal. The peas were either yellow or green, not a blend, not purple, yellow or green. The seeds were either round or wrinkled, and some people and some texts say smooth versus wrinkled. Then the pea pods themselves, round or wrinkled peas could be in inflated or constricted pods, and you could potentially have yellow seeds in green pods. So pod color was inherited separately from pod shape and from seed shape. And then the last one is stem length. Some of his plants were short and some were tall. There wasn't an intermediate height. It was just tall or short, and he called the short dwarf. And if you look at these ratios here, he counted, somewhere in the neighborhood of 8,000 peas. And when he was, and this is just the ones that he counted in what we call the F2 generation, and I'll get to that in a minute. But what this chart shows is a 3-to-1 ratio of purple to white flower colors, axial to terminal flowers, yellow to greens pea seeds, round or smooth to wrinkled, inflated to constricted, green to yellow pods, and tall to dwarf plants. And this has to do with crossing parent pea plants, then getting a generation of seeds, and then replanting those and growing those, and then getting the seeds from those and growing those and counting what happened. So this is from like the 3rd generation, which we call the F2 generation. And the text and this PowerPoint focus on just one of the characters, what he did with the flowers, the purple and the white flowers. So what he had in his parent generation was plants that always grew, they always gave seeds that grew to purple flowering plants, or always grew seeds that grew to white flowering plants. And he called these true breeding. meaning it's only going to give what it is itself. And so if he kept those purple flowering pea plants isolated from the white ones, every generation he was going to get purple. And if he kept the white flowering pea plants isolated from the purple, every generation he was going to get white. So he called them true breeding because they always gave offspring like what they were themselves. So then what did he decide to do with them? Well, what happens if I cross-pollinate the purple and the yellow and the white flowers with each other? And so then when the flowers are fertilized and the seeds grow, and then I plant the seeds, what kind of flower color am I going to get? And he called this the first filial or first offspring generation of this cross of the true breeding parents. And what did he get? Every single seed that he planted grew a purple flowering plant. Well, where did the white flower color go? It was gone. So the hybrids, the cross between the true breeding parents, only showed purple flower color. What does that mean about the gene that codes for flower color? There are two versions of that gene, two alleles, one for purple, one for white. Did the white one like get eaten up in the embryo somewhere, or is it just not being expressed, shown visibly? It's the latter. So he said that purple flower color was dominant to white. This is where the terms dominant and recessive come from when we talk about genes and traits that are inherited. It was coined by Gregor Mendel. So then he took that first offspring generation and he, this shows, hey, crossing the parents, and then the F1 generation, the first offspring, all had purple flowers. So the white appeared to disappear. But then what happened when he took those, these guys right here, this first generation, and he took them and he cross-pollinated, and then he took the seeds that were grown and he planted them. Do you know what color flowers he got? He got 3/4 purple and 1/4 white. And so what he knew is that the white didn't disappear forever. It was just hidden, and it's what he called recessive. And so he knew for it to show up when the F1 generation was crossed by itself, self-pollinated, that, that purple and white had to be inherited separately. And this is what he called his law of segregation. And he basically discovered that the ratio, when you cross true breeding plants and then you cross the F1 generation times itself, when you self-pollinate, gives you that 3-to-1 ratio. And if I back up a little bit, what did he find with all 7 of his traits when he did that? Do you see this average ratio here? He found with every single one of them that there was a 3-to-1 ratio. And so what did he decide about purple versus white? The purple allele, the purple version of the flower color gene, is dominant to white. The axial flower position gene or allele is dominant to terminal. Yellow seed color is dominant to green seed color. Smooth or round seed shape is dominant to wrinkled. Inflated pod shape is dominant to constricted. Green pod color is dominant to yellow, which is kind of funky because it's the opposite for seed color, right? And that 3/4 of the time he was going to get, oh, sorry. He was going to get tall plants compared to short plants. Any questions so far?
No.
No? Okay. All right. So moving forward again, he doesn't know about the separation of homologous chromosome pairs during meiosis. But that is why the purple and the white were inherited separately, because what happens to the parental chromosomes? They get separated and they're in individual eggs and sperm. And then when they come back together, a purple with a white is still going to lead to purple because the purple gets expressed dominantly over the white. If a white and a white come together, then you're going to get a white flower. And if a purple and a purple come together, then you're going to get a purple flower. And what he called a heritable factor is what we now identify as a gene, a length of DNA on a chromosome that codes for a specific protein. And in this case, it's a protein that determines flower petal color. Okay, so his hypothesis about this 3-to-1 inheritance pattern that he saw with all 7 of those characters is what we look at for the law of segregation. And so there's 4 pieces to it. And the first one is that there are alternate versions of genes. Well, purple's different than white. So it's not that there's one gene that codes for purple and one gene that codes for white. It's that there's one gene and you get 2 copies of each gene, right? One from either parent, and they're either the same or they're different. And if they're the same, then they're what we call homozygous, homo being the same. And if they're different, they're heterozygous, hetero meaning different, okay? So the gene for flower color, there were 2 versions, one for purple and one for white. And what he called them as heritable factors, we now call alleles. And if you look at this diagram, it shows you, hey, on a particular chromosome, there's a length of DNA that codes for the protein that gives the pea flowers their color. And in a homologous pair, if this is a heterozygous individual, then it might have purple flowers, right? But it has an allele that codes for purple and an allele that codes for white. And it's this allele that codes for purple, it's that protein that's visibly expressed. And so we say that the phenotype, the visible expression, is purple over white. So alternate versions, they're alleles. The second part is that one of each comes from a different parent. You don't get 2 from one parent and none from the other. You get one version of each gene, one allele from each parent. And he didn't know about chromosomes when he came up with it. So his idea got validated later. And basically, if it's a true breeding plant or a true breeding whatever it is, not looking at pea plants, then the alleles would be identical. And that's where we say they are homozygous or the same. And if they're different, then they're heterozygous. Okay, this is a kind of fuzzy example, and it doesn't show dominant and recessive, but if you look at the parent aliens, they each have 1 horn. So they have an allele. So on the chromosome where the gene is located, each of the parents would have an allele that codes for growing a horn and an allele that doesn't. And when you look at their possible offspring, if they each give either one or the other, their gametes are on the outside, and so they could have a chance of having an offspring. And actually here, the HH, the big H leads to no horns, and getting 2 little H's leads to 2 horns, and then just getting 1 leads to having 1 horn. So this isn't dominant or recessive, but it actually shows 1 allele from each parent pretty clearly, okay?
So does that mean though that like the one with no horn doesn't have the gene with the horn in it?
No, it means that he has a gene where the protein is different. Like the protein, the protein once it's made doesn't actually cause the production of horns. So it's like whatever allele this is and the protein that's made, maybe it's a dysfunctional mutated protein, but it's nonetheless a protein that's coded for. Okay. We're gonna get to codominance in a little bit, or incomplete dominance, and this is just kind of something interesting that it gets double. The only thing that this is meant to show is that 1 allele comes from each parent. You don't get both.
I guess my question is like, what makes a recessive gene be expressed? Okay,
so to get a recessive gene be expressed, you actually have to have 2 copies of the recessive. But it
still won't be expressed every time, right?
Well, here's an example. Most people you know are 5-fingered, right?
Yeah.
5 fingers is actually a recessive version of the allele that codes for the number of digits, fingers and toes, okay? And rarely somebody has 1 copy or maybe 2 copies of the dominant version. And whether they have 1 or 2 copies, if they have the dominant version of the gene, they have some sort of 6th finger and toe digit on their hand and feet, often surgically removed. And so the allele that codes for 6 fingers is dominant, but most of the population has 2 recessive copies. And so most of us are 5-fingered because all we can give is 5 fingers. And if I marry and have a baby with somebody with 5 fingers who didn't have one surgically removed, then all we can give is the recessive version. And so all of our children, short of a new fresh mutation, would have 5 fingers. So recessive just means you have to have 2 copies in order for the trait to be expressed. You have to be homozygous. If you're dominant, you only need 1 copy to express that particular allele version, okay? So to get white flower color, you have to have 2 white flower color alleles. To be purple, 1 will do, because the 1 will be dominant over the other. Purple will be dominant over the
It's like
white.
brown eyes and blue eyes. If you want blue eyes, you both have to have blue eye genes, but you don't both have to have brown eye genes.
Yeah, although eye color is a little more complicated than that. There's more than 1 gene and more than 1 allele.
Okay.
Because they're not just brown-eyed and blue-eyed people. My eyes are hazel, trending toward green. Yeah, so eye color is sort of a funky example, but the fingers really make sense to people, because how many people do you know with 6 fingers?
Right.
Right.
Okay. Thank you.
Yep. So the third idea is dominant over recessive. Okay. So Mendelian nailed dominance and recessiveness. He left out all the complicated stuff that he couldn't explain that we've subsequently tried to explain. So, you know, the first half of this is just figuring out the law of segregation, dominant versus recessive, okay? And the second half is all the ways in which genes depart from Mendelian dominant versus recessive. He looked at deliberately 7 pea plants that were inherited separately. I bet you can guess it gets way more complicated than that. Okay, so the fourth part of the law of segregation just explains what we know based on meiosis, that the 2 alleles separate during gamete formation and end up in different gametes. So an egg or a sperm only gets 1 copy from the parent. It doesn't get both copies. And so that segregation of alleles is based on separation of homologous chromosomes in meiosis 1, okay? So here's a human example. If a dwarf individual and a normal individual have a child, what is the chance that their child will have normal height or will have the dwarf height be expressed, okay? Now this is looking at if the dwarf was a dwarf child of a dwarf and a normal parent. If the dwarf themselves had dwarf parents, it would be sort of interesting if you could get 2 dominant dwarf genes. So the gene for dwarfism, for short stature and for some of the other things that goes with it, if it's inherited, the allele that codes for dwarfism, for the stature and the other characteristics, is dominant. So all you need is 1 copy. So if you look at the parents, a normal height person and the dwarf, then the dwarf can give either the dominant allele or the recessive allele, right? And the normal stature parent can only give the normal stature recessive allele. And then, so what happens here is there's a 50% chance, whether you're male or female, to receive the dominant dwarf gene. So approximately half of the children produced by this couple would be expected to show the dwarf traits. I have some friends who are both normal height, and their daughter's dwarfism is actually a new mutation, not a heritable thing, but there is heritable dwarfism. I knew a family of 5 girls in high school, and one of them was really short anyway. She was like 4'10", and she married somebody who genetically was a dwarf, and so their children were all going to be pretty short anyway, but half of them would be expected to genetically have dwarfism, and then half would be expected to be normal height. Okay, so his law of segregation that he came up with basically explains the 3-to-1 ratio, and it's all based on how alleles are inherited through the process of meiosis. Okay, so how can we predict what's going to happen? We can do something called a Punnett square. So you have a homework assignment. The second homework assignment that's due next week is on Punnett squares, and my Zoom on Thursday is going to be about Punnett squares. Okay.
I love Punnett squares. I'm so good at them.
Good. So basically, you know, looking at this just recaps. If you have the true breeding, the first generation is all purple and heterozygous because of one allele from each true breeding parent. And then what are the gametes that those flowers can make? What are the possible eggs and sperm from that heterozygous purple flower? Well, it could either give big P or little p, and therefore its eggs and its sperm, if it's going to self-pollinate, and so you can have pea plants do this where you can't have people self-fertilize, right? But a flower of a pea plant will make both sperm and eggs, and so you can have it self-pollinate. And it's either going to make its sperm and its eggs are going to be 50-50. And remember, because the zygotes are represented inside, because the gametes are haploid, they either get the dominant allele or the recessive allele. And so you can see we can predict that 3/4 of the offspring would have at least one dominant allele and therefore have purple flowers. And you can make the same Punnett square prediction for all 7 of the pea plant characters that Mendel investigated. All right, so we've talked through this vocabulary. Genotype is what your genes code for. Phenotype is what you look like. So you can see that whether the flowers were homozygous dominant, big P, or heterozygous, big P, little p, what was the phenotype? What was the visible expression of those genes? Purple flowers. And only if you were a homozygous recessive, little p, would you show the recessive phenotype of white flowers. So you have some purple flowered pea plants. How do you tell what they are? This slide says, well, hey, if we take this purple flowering plant and we don't know what it is, we don't know if it's true breeding, how do we test it? We test it against the recessive phenotype, because we know the recessive phenotype has 2 of the same allele. And so we know that the recessive one is only going to give the little p to sperm or eggs if we flipped it the other way, right? And if this purple flowering plant is true breeding homozygous dominant, then its eggs would be big P, and all the offspring would be purple. If it is a heterozygote and we cross it with a recessive individual, then half of its eggs or half of its sperm, we could flip eggs and sperm here, would be dominant and half would be recessive. And so then we would expect half of the offspring to show the dominant trait and half to be recessive. So if you cross 2 individuals and one of them is a homozygous recessive, if you get any offspring with the homozygous recessive trait, then what do you know about the unknown parent? That it was a heterozygote. The second law, the law of independent assortment, is basically looking at single characters. What we were just looking at as a Punnett square was what we call a monohybrid cross. And you are not going to have to do dihybrid crosses on the test, but I want you to understand how we used them to come up with the law of segregation. So basically, what happens if he takes seeds from a plant that are true breeding for two different characters. In this case, seeds that were yellow and smooth or round, and green and wrinkled. So green and wrinkled is the homozygous recessive trait. And if he crosses those, if they travel together, if they're essentially on the same chromosome, then he should never get any yellow wrinkled seeds or any green round smooth seeds. You would expect what's here on the left, that you wouldn't get any intermediates. But if in fact they are on separate chromosomes and inherited independently, then you get this interesting combination here of the dihybrid cross, di meaning two, hybrid meaning we're crossing and making a hybrid of these true breeding individuals. And look at what he actually got, was this table on the right. And if you look at the yellow, can you see that 3/4 of the offspring were yellow and 1/4 were green? If you count smooth or round, can you see that there were only 4 wrinkled seeds? 4 out of 16 or 1 out of 4, and 12 out of 16 or 3/4 were round. So the 3-to-1 ratio for round to wrinkled and for yellow to green holds up, but look at what you actually see. There's a little bit of a recombining where, because the gene for texture, smooth versus wrinkled, is on a different chromosome than yellow versus green. So the offspring could be either yellow and round, or green and round, or yellow and wrinkled, or green and wrinkled. And so the phenotypic ratio that is 9:3 to 3:1 in a dihybrid cross is basically the evidence that he had for what we now know is independent assortment of homologous pairs at the metaphase plate in meiosis 1. That's what it's evidence of. You aren't going to have to construct a dihybrid cross, but you need to understand what the 9:3 to 3:1 ratio means. Okay, so this just, the next couple of slides review probability, and there's some great stuff about Punnett squares and dihybrid crosses in the videos that I gave you for chapter 8. And the last thing I want to do is tell you the rest of the chapter is about non-Mendelian genetics. So this first table or row in the table is about complete dominance of one allele, but there are many ways in which things are not truly dominant or recessive. So you'll read about incomplete dominance, codominance, multiple alleles, pleiotropy, epistasis, and polygenic inheritance. And then the last thing that is in the PowerPoint, past polygenic inheritance, is a little bit about environmental influence and then about sex-linked traits. Those aren't actually in the table. And so what I want to have you do is read chapter 8 and watch a couple of videos and get that first assignment done. And if you struggle with it, then we can go over it on Thursday. And so just a reminder, here is the assignment. And it actually walks you through understanding ratios and probability by counting first shapes and boxes, and then flipping heads and tails, looking at Mendel's example, and then actually practicing with, I don't know, are these beetles or sheep? Okay, beetles. And then blocks in bags. And so it just kind of walks you through probability and helps you understand it a lot better. And so I can pick up with this on Thursday when we meet.
I think it'd be cool if you tried to stay up the whole time between